Good afternoon, everyone. A very warm welcome to all of you to the ANRF webinar series. It is my great pleasure to introduce our distinguished speaker for the today's session, Professor Dr. Jayachandran K. Jayachandran K is a senior professor at the School of Biosciences, Mahatma Gandhi University, Kottayam, with over 27 years of teaching and research experience in the field of bioprocess technology. Throughout his illustrious academic career, he has made significant contributions to the microbial biotechnology, bioremediation, environmental biotechnology, industrial microbiology, metabolomics, enzyme technology, and plant microbe interactions. He obtained his MSc in biotechnology and later completed his PhD from Cochin University of Science and Technology. Professor Jayachandran has successfully guided 15 PhD scholars and numerous postgraduate research students while authoring more than 85 research publications, several book chapters, and edited books in reputed national and international journals and publishing houses. His innovative research has also led to the award of two Indian patents, underscoring his commitment to translating scientific discoveries into practical applications. In addition to his research accomplishments, he has held several key leadership positions at Mahatma Gandhi University, including Registrar in Charge, Director of Research, Director of Graduate School, and Honorary Director of the Inter University Center for Studies in Science of Music. He has also served as a member of various academic and administrative bodies, contributing significantly to higher education and research governance. Today, we are privileged to have such an accomplished scientist and academician with us to share his insights. Please join me in extending a warm welcome to Professor Dr. Jayachandran K. Sir, the virtual stage is now yours.
Thank you, Ashwati, for your nice words of introduction. So, I hope I am audible to you.
Yes, sir.
Okay, thank you. So, let us straight away start our business. Let me share this slide now. I hope the slide is visible.
Yes, sir.
Okay. So, good afternoon once again. I'm here to initiate a discussion on the topic of our ANRF project. Now, the topic is biomarker identification and development of rapid low-cost marker testing methods using biological fluids such as serum, saliva, urine, and nasal secretion for the detection of fatty liver and diabetes. So let me put in a very short way. It is actually concerned, the project is concerned with development of marker-based rapid testing methods for fatty liver and diabetes. So thank you very much to ANRF for supporting this work, and big thanks to Hyderabad, University of Hyderabad, and special thanks to Dr. VVSS Srikant, our mentor. And see, I express my sincere thanks to our principal investigator, Professor Dr. Jayachandran K, and also to the ANRF fellow of this project, Ashwati KV. Now, to start with, what is fatty liver? I'm sure no, we had so many other presentations earlier. They might have also explained, might have started with fatty liver. Now, the fatty liver is a very common problem. in the public right now. Technically, we say if the liver is having a weight of more than 5% is of fat accumulation, then we say it is a fatty liver. Very common it is, and more than that, uh, there is growing public concern over this. Now, if you just get into fatty liver, we know we can classify that into different categories. Um, Very common one is alcoholic fatty liver disease, which is rather caused by excessive alcohol consumption. And another one is non-alcoholic fatty liver disease. Um, so this is the topic which we are right now concerned with. I'm sorry. Now, even when we say non-alcoholic fatty liver disease, we have the current terminology as MASLD, metabolic dysfunction associated steatotic liver disease. So hereafter we will be referring to MASLD, not to the AFLD. So what is this MASLD? Just for the sake of introducing the topic, I think we will just glance through this. We can see the normal liver where the fat is less than 5% of the weight of the liver, and the conversion of this healthy situation to the second stage, we start calling that second stage as MASLD. So this is actually a situation where there exists reversible chance of reversible process that this situation can be reversed if you are careful enough. And then the third stage comes, MASH, where we can see much more damaging taking place to liver. And then the other stage, cirrhosis, liver cirrhosis, very, I should say, threatening stage. There is no scope of a reversible reaction in this case. And ultimately, it may land up with hepatocellular carcinoma. And now there is, you can see in so many cases, liver transplantation is tried, but not that much successful, that is. So this is actually the situation. MASLD, when we say it is very common, when we say there is public concern, at the same time, it is threatening. It is not a very simple thing. So we are concerned with this MASLD. At the same time, there is an unfortunate part for this. MASLD and type 2 diabetes, we know the importance of type 2 diabetes. Now it's very difficult to find out a person after 40 without type 2 diabetes. So that is the common situation now. So when you think of type 2 diabetes and fatty liver, these two things we can see as coexisting. MASLD is usually found associated with type 2 diabetes. So then when this situation comes, things will become more dangerous. Why? Because there is a bidirectional link between these two, MASLD and type 2 diabetes. Approximately 70 to 80% of the people with type 2 diabetes, they may also develop fatty liver. They may also develop MASLD. Diabetes can give way to fatty liver. At the same time, fatty liver can give rise to diabetes also. So this combination is more threatening than the individual existence. So we are really concerned with this situation. Our project sharply focuses on this situation in particular, the cases where we have MASLD and type 2 diabetes existing together. Another important concern about this situation is that it is not easily detectable at early stage. So when it becomes advanced, then only people go and get it detected. So early detection is normally not taking place. People may not recognize that this kind of situation is going on in their body. Very rarely one gets the feeling of that. So that is the worst part of this combination. Both those things. Individually, you take individually MASLD, you take type 2 diabetes. In individual case also, it is very difficult to detect at early stage. When it is in combination also, there exists that kind of a challenge. So here, I have stated the problem right now, but what is the solution? Now we have to think in terms of solution. So this project focuses on that solution. What can be the solution for this alarming situation? The solution is to develop a rapid detection method. Doesn't mean that there are no such methods existing right now. There are methods existing. So many methods are existing. Whether we can, enhance this, whether we can increase the efficiency of rapid detection, whether we can increase the stability of the detection. Now that is now the question. So we are thinking about that solution. How far we can contribute to rapid, reliable detection. That will definitely help in preventing the unexpected or untimely heart event, strokes, kidney disease, or any kind of cirrhosis. Now, if you just analyze the present situation, this is happening throughout. Now, in particular in Kerala, you can see untimely event of heart attacks, strokes, kidney disease, and cirrhosis. So we are on the solution part of this. And one positive aspect which we have to recognize, if at all if you are able to find out this possibility at an early stage, why you are so hopeful. Now the positive thing is that just by managing lifestyle, just by managing your daily behavior, you can one way or other way manage this situation. So that is why we in this project focused on this combination of MASLD and type 2 diabetes. And our objective is to find out suitable biomarker for this detection. Why this is important, and we know many metabolic activities are taking place in our body. Maybe focused on proteins, maybe focusing on lipids, or something else. So large number of reactions are taking place, and we have the mitochondria as the powerhouse of the cell, where we have immense number of enzymes active throughout. So these metabolic reactions create so many products, intermediates, final products, everything. So if you can carefully go through these metabolic events, and if you can characterize the different compounds during the various pathways, that will be of great advantage. So you can classify these molecules, and you can evaluate the possibility of selecting these as a biomarker. So this is not the first time this kind of an objective is taken into consideration. We have several examples already in the literature, where you can see number of metabolites being taken as markers. Now, in this case, what we are trying to do, whether we can get marker for both this, when type 2 diabetes is happening, there are markers available. So many metabolites are formed, and when this MASLD is happening, so many metabolites are formed. So we will also look into that possibility of selecting one single biomarker, which will be very able to reflect the coexistence of these two. So these are the fundamentals, or I would say the platform on which our project is about to take off. Now, when you say marker, selection of marker through metabolomics, so that's what I have mentioned. There are many works going on, And there are possibilities. We can concentrate on these lipid metabolites. We can concentrate on amino acid metabolites, bile acid metabolites. We can concentrate on carbohydrate related products. We can concentrate on oxidative stress. So this kind of analysis can be done with mass spectrometry uh, using different samples, and we can classify the metabolites and we can look into the possibility of developing as a marker. No, what is your ultimate planning? That is also very important. But planning is to develop a biosensor based on the marker selected. So we have to select the marker at first, and then we have to design the biosensor. Biosensor, as we all know, it's very normally we say biosensor is developed by it's a marriage between biochemistry and molecular biology, we can say, or chemistry, we can say, or we can say electronics, we can say. So we need a biological component for that. The biological component will be reacting with the analyte. So when there is a biological reaction, energy difference will be there, substrate change will be there. So that change will be taken into consideration, and the transducer element will convert that change into another form of change, maybe into respective electrical change, maybe into respective temperature change, maybe into the respective optical activity change. So we need a biological component which we call as maybe a bioreceptor, for designing the biosensor, and then we need a transducer, and then whatever change is brought in, that should be amplified, detected, and recorded. So we have the processor and the reader. So these are the three elements, and more than that, one thing we have to be very careful. Even when you start with a bioreceptor or biological component, the component has to get the target at its immediate vicinity. So there will be so many metabolites in the sample. Only the selected targets should get into the system and react with the receptor or biological component. For that, we need membrane with absolute specificity. So membrane designing is also a part of that. So once we select a suitable biomarker, we have to get into the possibility of the biological component, and then we have to select the transducer, and we have to go with the processor and the reader. So this is actually what I was talking about. This is the target. So target reacts with the biological component. That reaction will create some energy change, which will become converted to another form by the transducer, and it will be amplified, and then it is recorded. So we can have many types of mechanisms in the selection of the transducer. Maybe conductimetric in the sense the produced charge species, how its conductivity varies, that be taken into account. You can have the amperometric biosensor, which depends upon the magnitude of the current. We can have the potentiometric biosensor, which is rather there are so many examples for that. We can use field effect transistors, piezoelectric crystals, another possibility. We have biosensors made up of that. We can make use of thermistors, optoelectronics, surface plasma resonance, normally named as SPRs, very common now. We can have a redox mediated system. We can have aptamers. So once the marker is selected, once the mechanism of identification is selected, we have to select the transducer system from this choice. As far as a present project is concerned, we will be mostly focusing on these possibilities, whether we will be able to select a target and a transducer system, either from conductimetric or amperometric or potentiometric. So this is actually the aim, to identify a reliable marker and to define a suitable detection system. Now, even after getting those things done, we will be focusing on the selectivity. If you have a marker, if you select a marker and if you design a transducer, and then you can even go for standardizing the whole process of detection. How much selectivity is there for your membrane? Well, that can be changed. Membrane can be modified to increase the selectivity. to increase the performance. And at the same time, you can just test whether your response is linear, whether your results are linear. So when you increase the concentration of the target, the response, whether it is increasing or not, or how far you keep linearity with that. So sensitivity is also very important. What is the minimum concentration which you can decide, which you can detect with your design sensor? Then stability, how far it is working, whether it is unstable, how much time you can use, and what is the detection limit. So normally when you have biosensor developed, detection limit will be very narrow. Like for example, maybe 10 micro to 100 micro, or 100 micro to 500 micro, something like that. So what is the detection limit? Now our attempt should be to extend the range of detection limit. So what is the response time? How much time it will be taken for detection and recovery time? If the developed biosensor is put into action, how much time you require for putting it into next action? So the recovery time. So once we decide, once we design the biosensor, we have to do the characterization of that. And we have the possibility right now for introducing the latest technologies into biosensor. We have so many examples. So we can also think of the next generation biosensors where we can have the application of AI into that. And then coming to the final objectives of the work, based on whatever we have discussed. See, this situation, like a combination of fatty liver and diabetes, we need to make a very extensive analysis in the public domain. We have to analyze the rural community, we have to analyze the urban population, we have to analyze what is the situation in the age below 20, what is happening above 20, what is happening above 50. So all such things we need to develop. We need to understand the influence of food habits into that. So we have to be very careful about all these things, because these are the facts. So our attempt should be exactly based on the existing facts. So those data collection is very important, and on that we have to work. So along with the data collection, we'll be taking samples, and the samples will be analyzed for suitable marker. Now the responsibility is to identify the marker. You'll be getting number of markers. Now the probable question will be how far you will be able to generate transducer for that. Even if we are getting, even when we receive number of biomarkers, we may not be able to develop transducer for all of them. So after selecting suitable biomarker, we have to screen those biomarker for suitability of biosensor development, like how far it is possible to apply embryometric sensor into that, or potentiometric biosensor into that. That will be a very huge question. And then comes the designing of the membrane with absolute specificity. So these are will be our major objectives, and we have already specified the inclusion criteria and the exclusion criteria. Like we will be depending upon 18 to 65 age, male and female. Then we'll be taking the family history, because as you know, the type 2 diabetes, and in this case also there is a genetic linkage, so that should be taken into account. And now the detection is very important, and we need ultrasound of FibroScan on one aspect for fatty liver, and we need the SBA1 test for type 2 diabetes also should be detected. And then we need a healthy group and control group. We need a group with obesity and willingness, all such things. We are ready for everything. Only requirement is the fund. Once we get the fund, I hope we will be able to do this business, this kind of initial, data collection, sample collection, and all such things. And exclusion criteria, obviously, those persons with disability or pregnant women will be eliminated, and those with viral diseases will be eliminated, and those who are taking immunosuppressant drug will be eliminated, and such things we have all scheduled now. So hopefully we'll be dividing the specimen into the sample, into the design will be divided into 4 groups, 1, 2, and 3. We are thinking of collecting 100 samples per group, MASLD with type 2, MASLD without type 2, type 2 without MASLD, and healthy control. Maybe ideal combination of the various groups. And we'll be extensively looking into the metabolomics of all these things by making use of LCMS. Mass spectrometry will be utilized. So this is the overall idea of the work. We will be starting with the MASLD type 2 diabetic cases will be selected during the sample collection. The biological samples will be collected, will be taken for mass spectrometry, will be analyzing metabolomics, and then based on the marker selection, we will go to the biosensor, and if there is any requirement, if there is possibility, we will incorporate AA also into that. And then this is our final aim, getting it easily done. And normally, people are reluctant to go to the hospital. We are not happy to consult a doctor at all the time. So even without consulting that, if you're doubtful about your own system, you can have an easy check whether you are this situation converging into your body. You can easily do that with our possible biosensor. So that is our ultimate aim. As far as the situation is concerned, now we have constituted the Human Ethical Committee and the Institutional Biosafety Committee. Tremendous support is being offered by University of Hyderabad. We went there. We were having very good discussion with the scientists over there, and our professor mentor himself was instrumental in giving many suggestions and advices to us. We have presented the entire picture before Kottayam Medical College, and those people are very much interested to support us. In a way, we have also good collaboration with our own IUCBR Center, which is an established medical research center. We have purchased almost everything. We have completed the whole purchase. Now we are waiting for the sample collection procedure. In fact, we got permission from the university to conduct a medical camp. Even the permission also we have received. So we need the financial support for the same. Once the financial support is there, we are ready to take off. So that is the situation. So thank you from my side, and thank you very much for your patience. If you have anything to add or any question, if you have, please ask. Thank you.
Thank you, sir, for the session. Now the session is open for discussion.
Good afternoon, sir. Can you hear?
Yeah, please.
Sir, this is Reema, your student.
Yes, I have seen you on the desktop. Thank you for your presence.
My work.
How are you? How are you? How are you now?
I am doing well. Okay, I'm going into different projects. Actually, my work was mainly focused on the development of biomarkers in Trivandrum population. I have been doing it as a CSIR research associate, and we have a paper regarding it. So my doubt is, you have told that you are working on serum, then urine samples or saliva. You would be looking into all the samples, or specifically you would be zeroing into a particular sample.
Yeah, we will be looking into all the samples right now. That is what we have planned. Our intention is very clear. We want to get marker which will reflect the presence of both these. So we don't know, and one thing, our final intention is to make the detection rapid and reliable. So we don't know in which context we'll be getting that. So we have to make extensive screening. I understand your question. You mean to say that the screening will be very extensive in this case, is it? That's what you are aiming.
Yes, sir.
Yes, I got it. Your observation is perfect, correct. But we have to take it up anyway. We were doing that.
Okay, thank you, sir.
So if your sample collection is not wide, finally you will be in trouble. So what we are planning, stage by stage, it should be perfect, because our objective is not that simple. It is something which
Okay,
is
I'm
very, I would say, is very great. I would say so. To get great things, you have to put pain also. That's it.
Okay, and another question, how will you, as you said about the sample collection, how will you select the population? Like, have you set up any criterion for selecting a
Yeah, I
particular
think I have explained that. We have
population?
set up, we have
Not
defined the
like that,
criteria.
by
We will be
socioeconomic
following that
class.
criteria.
Yes, sir. Okay. Okay, I know that you got the ethical clearance. I
No,
just just to
see,
understand.
the
Mm.
criteria is very important to confirm the consistency of the detection system. If at all we are developing one system, how far it consistent, that is very important. Like, see, that criteria we have to define. So, for example, if you are selecting one marker, whether that marker is applicable in different age groups, something like that is very important. So we have to do that.
Okay, sir. Just a word. Actually, during our sample collection, our ethical committee was more critical on selecting different strata, and we were asked to do sample collection on under a different strata, like from different strata we have to select. So, do you have such a plan, or just
Yes, we do have. Now, what we are planning to do, we will be collecting samples from the urban areas, from the rural areas. We will be collecting samples in different age groups, male, female, all possible combinations will be trying. That is what we have planned. We know it's a hectic business, but we will be doing that because the advantage is that we have several projects on this right now being conducted at the university. So even though one fellow is associated with one project, so we have Ashwati with this. Collectively, we have so many people, so we can make a big effort. So this sample collection can be applied for all the projects uh, inside the university, not for this alone. So we are very hopeful on that.
thank you, sir. A very best wish for
Thank
for
you,
the
thank you.
successful project.
I'm really happy to see you here, this platform,
Okay.
after many
Actually,
years.
I have
I
been
remember you very well.
doing.
You are a very good, talented, dedicated student at the time. You got
Okay, thank
CSAR
you, sir.
just after the course. Am I right?
Can you
Yes.
repeat the question? I haven't heard. Can you repeat?
Yes, I'm saying I remember you very well. You were very
Okay.
good. You were very good student. No, I remember on those days how you were performing. You
Okay,
got
thank you, sir.
CSAR just after the MSc course, isn't it? Along with that course, I think.
No, I took nearly after 2 years I got.
Oh,
I
okay.
said,
Anyway, I'm really, really happy to see you here in this platform.
okay, thank you, sir.
Okay.
Thank you. And any help from my side, and also I would also
Definitely,
like
it is
to
required.
collaborate
Ashwati,
also.
please note and please take the email and the number. Be in contact with. Be extremely useful because one thing. It's for the first time we were starting this kind of a work.
Yes, sir.
So project. So I request that.
Okay. Thank you. Thank
Thank
you,
you.
sir.
Thank you.
Okay.
Thank you very much. Any addition? Any suggestion? Any criticism? Anything? Please come forward. If not, we can conclude.
If
Ashwati,
there is any queries, please ask.
yes, you can add. Now, your suggestions will be accepted as most valuable contributions because we our intention is to get the target at any cost. So whatever suggestions you are putting, we will be very thankful to you for all those suggestions. So please come forward and put your suggestions. So one thing is very critical. I know I feel only ANRF fellows are here in this group, uh, and you may be involved in different projects and guided by different professors of the university. But you have to be very critical on all presentations. You can you can make a very good critical analysis on each slide even. So do that, and that will I would say That will only increase the scientific depth of the attempt. Be very cautious on each slide. You can raise many questions on each slide, in fact. So do that for all the presentations. So that will increase our knowledge, our investigative spirit. Do that rather than sitting here and attending this speech through online platform. Why not you be very critical on each slide? So that will be very positive. So if there is no question, Ashwaji, we will conclude this.
Yes, sir. If there is no further queries, let's wind up the session. So, thank you once again, sir, for the wonderful session, and thank you all for joining.
Thank you very much for your patience. Bye. All the best.
Thank you, sir.